Hello everyone, I am back with our new chapter. So today is going to be chapter 19. It's going to be on the topic of variation and natural selection. So let's begin on with the definitions first. So there are at least 7 important definitions that you must be knowing from this chapter according to your latest syllabus. The first one is mutation, which is a genetic change. Gene mutation, which is change in the base sequence of DNA. Natural selection, which is the greater chance of passing on of genes by the best adapted organisms. Adaptive feature, an inherited feature that helps an organism to survive and reproduce in its environment, or the inherited functional features of an organism to increase its fitness. Normally when they ask for the adaptive feature, we usually mention about this inherited feature that helps an organism to survive and reproduce in its environment. But sometimes certain questions, they actually it's not really related to the definition, but they usually ask in your MCQ about relating to the fitness actually. So we'll just highlight this and right, they usually ask this in the MCQ. Next one is fitness, the probability of an organism surviving and reproducing in the environment which it's found in. Variation, differences between individuals of the same species. Process of adaptation, the process resulting from natural selection by which populations become more suited to the environment over many generations. Right, so let's move on to exercise 19.1. Water hyacinth experiment. There is a tough magnification calculation in this exercise using a unit you may not be familiar with. Don't worry, you're not supposed to know this unit. The task is about being able to use the understanding of magnification and the information given to work out the answer to a problem. Alright, and the exercise would also help you to look carefully at data and make comparisons and to think about adaptations of plants that grow in water. Now let's move on reading the text over here. Water hyacinths are aquatic plants that originally came from Brazil but now grow in waterways in many tropical countries. They are sometimes used to clean up polluted water as they're able to take up pollutants such as heavy metals. An experiment carried out in China investigated differences in the structure of the leaf epidermis of water hyacinth plants grown in clean water and in polluted water. The diagram and table shows some of the results. So before we actually move on, the text that you guys see in this box over here, this whole thing, like this whole text you guys see here, it's basically about converting from millimeter to micrometers. And micrometers, you guys may not be familiar with that unit, but you'll get to know it very soon. So it's basically, I'll just write it over here for your reference. It's basically from millimeter to micrometer, right? So yeah, now this is how the upper epidermis of a water hyacinth leaf looks like in a polluted water. So if you see the table over here, the type of water, which is when the water is clean, the upper epidermis of the mean width of the stomatal pore is 4 micrometers, mean length of the guard cell is 7 micrometers, and mean number of stomata is 2.83 millimeters square. And then the lower epidermis of the water hyacinth, when it's in clean water, the mean width is the same, it's 4, guard cell length is same, 7 micrometers, but the stomata number is a bit higher compared to the upper epidermis when in clean water. If you see, this is 2, if you see, this is 2 and this is 3.32, so this is higher than the upper epidermis. And this one is polluted, so this is the upper. So in polluted water, the upper epidermis, the mean width is 3. So that means in clean water, the stomatal pore is higher than the stomatal pore in the polluted water. And the guard cell, sorry, it's lower, not higher. And the guard cell is also lower when it's in polluted water. And the mean number of stomata per millimeter square, it's almost the same, it's not much difference, but if you compare the polluted water, the lower epidermis is always higher than the upper epidermis. So what you can conclude is that the mean number of the stomata per mm square is actually higher in the lower epidermis in both clean and polluted water when compared to the upper epidermis of the water hyacinth in clean and polluted water. Okay, so let's move on to the first part of the question, which is from the data in the table, what is the mean length? The mean length of a guard cell in the upper epidermis of a water hyacinth leaf grown in polluted water. So these are the key terms: polluted water, mean length of guard cell in the upper epidermis, so it's going to be 5. If you write just 5, they're actually going to deduct your marks. They're asking you to write the length. So if the question doesn't give you the unit, then you have to write down the unit, or else you would not gain any marks even though your value is actually correct. So putting the unit, usually in biology they always give you the unit, but sometimes in paper 6 where it's supposed to do usually in paper 6 they don't really mention the micrometer unit, which is this is the micrometer unit. They don't really mention in the question paper in paper 6. It depends on which questions, but usually they don't mention. It's usually in the magnification question where you have to convert from millimeter to micrometer. And then part B, measure the length in millimeter of a guard cell in the diagram of the epidermis. Measure the length of a guard cell in the diagram of the epidermis in a water hyacinth leaf. So measure the length. So they want you to find the length. So this, right. So if you see over here, this is actually a guard cell, this is another guard cell, this is another guard cell, this is another guard cell, another guard cell. So what they want from you is you need to measure the length of this guard cell, and don't forget they want you to mention in millimeters. If you guys mention in centimeters, the value is going to be wrong because the question is already given to you in what format they want your answer to be in, so it must be millimeters. However, when you measure the ruler from here to here, it's usually in it's actually supposed to be from here to here, but it's just that I don't have the thing, so yeah, right. So from here to here you guys have to measure it, so it's actually going to be in centimeters, but you guys have to convert it into millimeters. Sometimes there could be a plus minus. It is not necessarily has to be 12 millimeter. It could be 11, it could be 13 as well. They give you like a negotiation capacity between like 11 or 13, plus 1 minus 1, right? So now we're done with part A. Now convert the answer in millimeter to an answer in micrometer. 1 millimeter is equal to 1000 micrometer. Now remember this, in your paper 6 for biology IGCSE, this is very important to know the measurement of this scaling from 1 millimeter is equal to 1000 micrometer. This is absolutely important for you to remember. Remember it. It's very important for your paper 6, because in your paper 6, the second part of the question, when they ask you to measure the magnification question, you have to convert the millimeter into micrometer, just like what you did over here. You measured this, you measured this in centimeters, you convert into millimeters, and that millimeters you have to convert into micrometers. This whole thing is worth about 3 marks, so you can't just lose that 3 marks just like that. And it's a good thing that they mention you here, but in your original exam question paper, they will never ever give you the scaling. You have to memorize it by heart. The 1 millimeter is equal to 1000 micrometers, so our guard cell measure 12 millimeters is going to be 12,000 micrometers, so you write it down as 12,000 micrometers, right? So let's move on to the next part now. Use the answer to A and your answer to B in micrometer above to calculate the magnification in the diagram. Write down the formula that you use and show you're working. So magnification is equal to length in diagram divided by real length. Another simple way to remember is I. If you draw the triangle, I am, like the I is on top, which is for image, and then A is for actual, which is on the left hand side on the bottom, and magnification is on the right hand side on the bottom. So it's very easy for you to remember the formula triangle, which is I am, right? So if you see, this is 12,000 micrometer divided by 5 micrometer. Basically you take the 12,000 micrometer from here, then take this. So length in diagram is 12,000 micrometer and the real length is 5, so the magnification is times 2,400, right? So now let's move on to part C. Explain how the results in the table for water hyacinth leaves grown in clean water indicate that this plant is adapted for growing in water. So let's read the answer together. The leaves have many stomata on the upper surface. If you see in the table over here, the leaves have many stomata. This is the stomata in the upper surface, stomata in the upper surface, right? This is not usually found in living plants, where most stomata are on the lower surface to reduce the rate at which the water vapor is lost through them. The lower surface out of the direct sunlight. Lower surface is out of direct sunlight and therefore cooler, reducing the rate of evaporation and diffusion. The water hyacinth leaves are at the surface of the water, so they don't need to conserve water, and having stomata on the upper surface allows them to absorb carbon dioxide easily from the air. So it's pretty much easy to understand how you have to explain. You basically have to explain from the table above, but you have to use your knowledge of like what difference does it make to the diffusion, evaporation, transpiration, what happens to these three rates when there is more stomata in the upper surface, in the upper epidermis than in the lower epidermis, because normally there's actually the stomata when it's in normal living plants, like plants that grow in roots. Okay, water hyacinth, they grow in water, but plants that grow in roots, there are a lot of stomata on the lower surface. But for water hyacinth, the stomata is on the upper epidermis, which is quite unusual. So this is like the whole explanation. So the water hyacinth leaves are at the surface of the water. So since they're at the surface, they don't have to conserve water, so that's why there's no need of the having a lot of guard cells in the lower epidermis, and it allows the stomata to absorb carbon dioxide easily from the air, right? So let's move on to part D. Compare the characteristics of the leaf epidermis of the plants growing in clean water with plants growing in polluted water. The stomatal pores of the plants growing in polluted water are 1 micrometer smaller than those in clean water. So the stomatal pores, and now they're emphasizing on stomatal pores. This is a stomatal pore, 1 micrometer difference. Clean water is 1 micrometer higher, polluted water 1 micrometer lower, right? The guard cells of the plants growing in polluted water. The guard cells of the plants growing in polluted water are 2 micrometers shorter than those in clean water. So the guard cells of the plants growing in polluted water are 2 micrometers shorter than those. So if you see the guard cells over here, there are 2 micrometers shorter than those in clean. This is 7, this is 5, so the difference is 2, right? And the mean number of the stomata on the upper surfaces of the leaves is the same in clean and polluted water. The mean number of stomata on the upper surfaces of the leaf is the same in clean and polluted water, meaning it's same, 4 and 4 in clean and 3 and 3 in polluted. This is what they mean by. And the mean number of stomata on the lower surfaces is a little higher in the plants grown in clean water than in those in the polluted water, meaning there is higher in the lower epidermis than in the lower epidermis of the polluted water. So basically in this question, since you compare the characteristic of leaf epidermis, you basically use this whole knowledge in the table, compare everything in the table, which one is high, which one is lower, which one is equal, which one is the same, right? But for this one, since I ask you to explain the as I mentioned in one of my first or second videos, I mentioned that when the question is asked about explain, you don't just explain what you see in the table. You have to explain analogy. What difference does it make to transpiration, evaporation, since it's related to plants? So there are 3 important things: evaporation, transpiration, and the diffusion of all the carbon dioxide, oxygen, water, and all that, right? So yeah, right. So I'll upload a second part of this. So yeah, stay tuned.